Hi everyone. In this video, we will learn about genetic polymorphisms, or we can also say genetic variation. So imagine you are a pharmacist taking care of warfarin MTech. So MTech is a medication therapy adherence clinic. So you are in charge of the patients that are receiving warfarin. So all of your patients are taking the same dose of warfarin, say like 5 milligram OD. And they come for a follow-up, and then you found that some of the patients does not reach the target iron out, showing that they have insufficient response to warfarin. And on the other hand, some of the patients are actually having bleeding events, which means that they might have an overdose of warfarin. But some of them are actually responding very well with the same dose, and they're not having any side effects or any bleeding events. So how do you explain that to your patients? So by the end of the lecture, you will be able to explain the terms commonly used in pharmacogenomics, like alleles, genotype, phenotype, haplotype, describe the definition and types of genetic polymorphisms, to explain Hardy-Weinberg principle, and to understand gene nomenclature and databases for genetic variation information. So a little bit of history here, since pharmacogenomics field is not new actually. As early as 510 BC, Pythagoras has recognized the dangers of eating fava beans for some people, and it was later associated with G6PD deficiency. So it shows that some people might show a reaction that are not experienced by others. In 1866, Mendel established the rule of heredity. And fast forward in 1957, Motulsky discovered the inheritance of defects in genes related to metabolism, which might explain why certain individuals have different drug response profile, which is already a big component of pharmacogenetics. Then Volkel has coined the term of pharmacogenetics. So pharmacogenetics concept has already been discovered in 1957. In 1988, variations in CYP2D6 is characterized, and following that, there are various polymorphisms in enzymes and proteins are further identified. Now let's look at some of the terms in pharmacogenomics. So what are genomics itself? So it is a study of a complete set of genetic information present in a cell or an organism or a species. Whereas genomic medicine is basically how the knowledge of genetic information were used as part of clinical care, for example, in diagnostic or the treatment of a disease. So it's a very general or broad term of using the genetic profile in medicine. Pharmacogenetics and pharmacogenomics. So what are the difference? Sometimes we can use both of these terms interchangeably. Definition-wise, pharmacogenetics is the study of variations of DNA and RNA characteristics as related to drug response. So we have the pharmaco component and the genetic components, which means we actually integrate that we relate that with the pharmacokinetics and pharmacodynamics of a drug. Which later we saw that as a drug response or drug toxicity. In pharmacogenomics, it is more broad. So it's a branch of science concerned with the identification of genetic attributes of an individual that lead to variable response to drug. So pharmacogenomics is more of a more broader definition, whereas pharmacogenetics is more specific to that study of a specific genetic variation that might lead to any specific drug response or drug toxicity. Human genes contain two alleles at a particular genetic position, or we call locus. So each allele are inherited from our parents. So both allele can either be coding for the same feature or a different feature. So for example here, short hair or long hair, so this is not for human actually, white fur or black fur. So each of these allele encodes for the different phenotype. So which color of fur do this animal get It depends on which allele is the dominant one. Polymorphism is a difference in DNA sequence. So note here that it is actually not a defect. So it is just a variation of DNA sequence among individuals, which can either be in the same population or ethnicity or in a different population. So each of us have a different genetic coding, a genetic sequence, and it's not necessarily be a mutation or a rare situation. It's just how we human varies. So the allele frequency of this different DNA sequence should be 1% or greater to be called as polymorphism or variation. Otherwise, if it's less than 1%, um, it, it will be called as mutation. So for example, in 100 students in this class, 3 of you have alleles of brown eyes. So 3% of this class has this polymorphism causing a phenotype of brown eyes. So what about genotype? So it is a combination of two allelic variants of a gene in a given individual. So everyone has their own genotype for each allele. For example, here we have two alleles, one each from maternal and paternal side. And for each genetic locus, if we read the nucleotide base here, from the maternal side we have G, and for the paternal side we have T. So the genotype here, we just read GT, as shown here, GT. Haplotype. So haplotype is a set of DNA polymorphisms, or usually referred to as SNPs. They are always inherited together. So if you look at the example here of this gene, there are 4 SNPs at different locations, 1, 2, 3, and 4. We read them as genotype for each polymorphism, or we can read them as haplotype of 4 polymorphisms. So this example has 3 haplotypes in which consisting of different combination of alleles of these 4 variations. So don't worry too much about this haplotype. It's more important for you to know about the genotype at this stage at least. So phenotype, so what is phenotype? So it is actually the observable characteristic of an organism, in which it means that the feature that we have, or the characteristic that we have and we can see that are portrayed. So it can either be appearance, development, or behavior of the person, and which is determined by its genotype. For example, this person have a genotype of a brown hair color, and then the phenotype is brown hair. So now we will go to the types of genetic variation. I list here 5 of them. There might be more, but we'll only look at the most relevant and frequently observed variation. So the most popular one is SNPs, or we call it a single nucleotide polymorphism. As the name suggests, there is a polymorphism at a single nucleotide base at a particular gene locus. For example, here, at this particular gene locus, a lot of people have C in this location, nucleotide piece of C in this location, but for some people, they might have T instead of C. So it occurs only at one nucleotide base. This is the example of a SNP, and this is how SNP is usually written. So this is the name of the gene ABCB1, which encodes for P-glycoprotein. So this is the name of the protein that it encodes. So this is the RS number of the SNP. So DB SNP is the registry of SNP. This is the SNP nomenclature, meaning that we have information of the location of the SNP, which is at the 3435 at the genetic locus, and C substituted by D and T. For example, a majority of the population has C, but instead some people have T, so it's C and T. This is the location of C. So instead of C, some people have T. So C here, we call it as reference allele, whereas T here, we call it as variant allele. So that means that there is a variation in this particular location. SNP is very common actually. It may occur every 100 to 300 bases along the 3 billion base human genome. So that makes up to 10 to 30 million of SNPs. But worry not all are functional or not all are relevant. SNP can either be synonymous or non-synonymous. Synonymous means that the SNP does not change the amino acid produced, whereas non-synonymous SNP are actually causing a change in the amino acid produced. So we will look further. For example, here we have two strands of chromosome at this location of the allele. So now we want to read the genotype of this SNP. So now we have C as a reference allele and T as the variant allele. So a person can either be a homozygous, which means that they have two of the same allele. For example, here CC, so they can have a C for in both strand of DNA, but they can also have a C in one chromosome and T in another one, and they can also have TT. So we call them as so if they are having CC, which is the wild type allele, so we call them as having homozygous wild type. Whereas if they have CT, they are called heterozygous, and if they have TT, we call them as homozygous variant. So genotype here is the CC, CT, or TT. So some more example, individual A, the first chromosome, we look at the upper one. So we have in this location T, and the second chromosome we have C. So the genotype for this person is TC. Whereas individual 2, we have C here in the first chromosome, and second chromosome is also C. So we call it as having a genotype of CC. So depending on which are the reference allele, for example, here is T is the reference allele. So that means that this patient is having heterozygous genotype, whereas in this individual, it has a homozygous variant genotype. Now we come to the second genetic variation, which is the insertion and deletion. So it occurs when a specific nucleotide sequence is present, which means it is inserted, or absent, which means that it is deleted from that DNA sequence. For example, here, the first sequence is in this location is CACAGG, but another individual, for example, the DNA strand had T in the middle of C and A. So that means this patient have a genetic variation of insertion. Whereas in this location, for example, it's a TGAG, but in the different individuals, the A, nucleotide base is deleted or absent. So this is called the deletion. So now we want to look at the consequences of these genetic variations. How would it affect protein produced? So for example here, if you look at the wild type, we have UUU translated into phenylalanine, for example. So imagine there is an addition of one nucleotide base into the strand, or a deletion of one nucleotide base from the strand. So it will shift the sequence forward or backward. So the codon are also changed. For example, here there is a deletion of U. So from UUU in this codon, it becomes UUG. So the amino acid that are translated is also different from PHE to LEU. Whereas if there is an addition of nucleotide base, instead of here AAG, now there is an addition of U, it becomes UAA. So it is a stop codon. So the translation process has stopped. Frame shift mutation is a term for the shifting of a location of nucleotide bases for each codon. So it is generally what we have discussed earlier. So for example here, there is an insertion. So for example, there is an insertion here. Instead of UAC, it becomes UA, and now the amino acid translated is also different. Where else, another example for deletion. So in this location, UAC, now C is deleted, so G is shift backwards. So the codon now is UAG, which is a stop codon. So now we look at the third genetic variation, which is the copy number variation or gene duplication. So for copy number variance or gene duplication, it occurs when the number of copies of a particular genetic sequence is different from the reference genome. Some copy number variants are not actually linked with any specific phenotype or any specific diseases, but there are as many as 40 that have been definitely linked with diseases. For example, genetic duplication of chromosome 21 is causing that person to have Down syndrome. So if we see the length of the chromosome itself, it will be extended as compared to the reference genome. The fourth genetic variation is variable number of tandem repeats. So VNTR occurs when a specific region of a DNA is being repeated in tandem, in variable number of times in tandem. We have a variable number of tandem repeats, or we have short tandem repeats. So for VNTR, you can see here, for example, there is one segment of the DNA that are repeated. For VNTR, it is 10 to 100 base pairs being repeated, whereas for the short tandem repeat, it is much shorter. So the segment that are repeated is from 2 to 9 base pairs. So the length of the genetic sequence is also different. When the person having this VNTR, it will be longer in length as compared to the reference genome. So this is the example of a short tandem repeat polymorphism. For example, in individual 3, so we can see that there is a repetitive unit here. For example, in this the first chromosome, so this region is repeated 3 times, whereas in the second chromosome it is repeated for 5 times. So the relevance of this repeat region is that when these specific repeat units are actually the codon that are encoding for certain proteins or certain amino acid, so the expression of that amino acid or that protein is higher than the person who do not have this repeat. The last one is the gene arrangement. So there is a structural alteration of the chromosome that causes a change in the order of its loci. For example, here in this location of the chromosome, it consists of two genes here, TPM3 and NTRK1, for example. And suddenly there is a DNA breakage here for this segment, and this segment is flipped, and then it rejoined with the rest of the chromosome again. So the order of the loci has already different from the original loci in other humans. So these are the more details of the example of chromosome or gene rearrangement. For example, chromosome inversion, chromosome translocation, or gene amplification. So let's recap a bit. So we have 2 strands of allele here from paternal side and maternal side. So if we take the example of this one location, so there is G here from paternal side and also G from maternal side. So this is the homozygous SNP. This is the homozygous genotype of GG. Allele frequency of the population says that the G allele is represented by 51% of the population, while T is represented by 49% of the population. So that means that there is a variation there, but only that in this person, he was having homozygous wild type. So we call G as the wild type, while T is the variant allele or minor allele. Whereas in the second position, we have A in the paternal side and C from maternal side. We have AC. So it is straight away, it is a heterozygous. And we know that it is a SNP because 90% of the population has A in this position, while 10% has C. So C here is the minor allele and A here is the wild type allele. So another exercise here, if we see here that the DNA and the mRNA strand, this is the normal situation or the normal allele. For this one, we can see a substitution from C to T here. So it caused an mRNA change also from G to A. So that means the codon is also changed. So the amino acid is also changed. So there is a substitution here at the location of C. So it is a SNP, single nucleotide polymorphism. Whereas in this strand, there is an addition of C in this particular region. So the codon is also shift. So there is a frame shift mutation happening here. So instead of CTT, it becomes CCT, and also the codon is different and the amino acid produced is different. So this is called the insertion, causing a frame shift mutation, and this is a missense mutation, meaning that from originally the amino acid is supposed to be GLU, but it becomes GLY. Another example is here when in this location it's supposed to be A, but in this strand A is deleted. So there is a frame shift mutation as well, in which T takes over A's place. So codon is also shift here. So the amino acid is also shift. So this is called deletion. So I think that's all for the first part of this genetic polymorphism video. And we will continue with the Hardy-Weinberg equation and gene nomenclature in the next video. Thank you.